Good morning, students. This is Veena teacher. Let us very quickly recapitulate what we saw in earlier module. In the previous module, we saw spontaneous mutations and induced mutations, gene mutation, chromosomal mutation, in which we have seen euploidy and aneuploidy, and we have also seen the significance of euploidy and aneuploidy. In today's module, we're going to start with genetic disorders. So there are 4 genetic disorders which are given in your syllabus. These are phenylketonuria, thalassemia, color blindness, and sickle cell anemia. Along with them, there are 3 chromosomal disorders: Down syndrome, Klinefelter syndrome, Turner syndrome, about which we are going to cover in our next module. So let us start with the first genetic disorder, that is PKU or phenylketonuria. If you remember, children, we have studied about this disease earlier also, but nevertheless, let me just quickly recapitulate or quickly discuss what we have seen about this particular disease. Phenylketonuria or PKU, it is caused due to the absence of the enzyme phenylalanine hydroxylase. This particular enzyme, that is phenylalanine hydroxylase, it is helpful in conversion of amino acid phenylalanine into tyrosine. So in the absence of this enzyme, the phenylalanine starts accumulating and it won't subsequently get converted into this amino acid tyrosine. The gene to produce this particular enzyme phenylalanine hydroxylase is present on the 12th chromosome, and it is an autosomal recessive disorder. So this particular gene is carried on 12th chromosome, which is your autosome, and this is a recessive disorder. Means if both the autosomes contain this defective gene, only then the person will show the symptoms of the disease. Now, accumulation of phenylalanine, it results in mental retardation in the newborn children. So this is about the disease phenylketonuria. Now let us go to the next disease. It is a very dangerous disease, even commonly seen also. This disease is known as thalassemia. I'm sure you might have heard about thalassemia. Now, before going into the disease details of thalassemia, let us first understand what is the structure of hemoglobin. In our blood, the hemoglobin is present, which contains 2 parts, heme part and globin part. Heme part is here, you can see the yellow colored structure, heme, iron containing pigment, and these loop structures or thread like structures is the protein structure, that is globin. So whatever food you take, if you take the food that is rich in iron, that contributes to the iron here, and then your body is having genes to produce this particular globin protein, which makes hemoglobin. The hemoglobin contains 4 polypeptide chains. 2 chains of alpha, here you can see alpha chain 1 and alpha chain 2, and 2 chains of beta, beta 1 and beta 2. So this is a structure of a normal healthy hemoglobin. Now let us see in thalassemia what the structure of hemoglobin, how the structure of hemoglobin looks in the patient of thalassemia. Now here in the patients of thalassemia, what can happen is one of the beta chains may not be produced. So if one beta chain is not produced, you can see the hemoglobin of that particular person looks something like this. And if both beta chains are not produced, then the hemoglobin structure looks something like this. So these persons are known as carriers, and these persons are the ones which suffer from the disease, and these people, they have severe anemia. In these individuals, severe anemia is seen. So this is the normal hemoglobin, no thalassemia. These individuals are carriers of hemophilia, though they are normal, but still they carry the gene that can produce the disease, and these individuals are thalassemic individuals. So when the marriage happens between 2 thalassemia people, that is when you can see that the children also can be born with this particular disease condition. So as you can see, the hemoglobin has 2 alpha chains and 2 beta globin chains, and their synthesis is controlled by 2 pairs of alpha globin and 1 pair of beta globin genes. So when a mutant gene is present, it is unable to synthesize normal alpha or beta chains. So what happens is the excess of globin is present, which accumulates in the RBC, and this leads to anemia. The person becomes extremely weak, and the only treatment that is available for this people is time to time blood transfusion. Now let us understand the next disease, that is known as sickle cell anemia. Now here in this picture you can see how the normal erythrocytes of the people look, and here you can see how the erythrocytes of the patient suffering with sickle cell anemia look. So these kinds of RBC molecule, this structure is known as sickle. So that's why you call this disease as sickle cell anemia. Now let us understand about this disease. Now here, what is the molecular basis of sickle cell anemia? Here in the normal, this is a particular gene, and here in this gene you are having these 3 nucleotides that are present in the DNA, and these 3 nucleotides are C, T and T. So in this particular T, this particular T is replaced by A. That is a time when the person will develop sickle cell anemia. So let us see what is happening when this replacement is taking place. Now when this is taking place, what exactly is happening is, this is the DNA, so this is the corresponding mRNA. Now all of you know that in the DNA, if the cytosine is present, in the mRNA it's going to be guanine, and here if thymine is present, correspondingly in the mRNA it's going to be the complementary base, that is adenine. And also here once again thymine is present, so that's the reason why here you have once again adenine. Now let us see how the mutant gene is present. In the mutant gene, the cytosine is present, so in the mRNA the guanine is present, that is normal. Whereas here the adenine is present, and here uracil is present. Children, at this point I want you to recall that in mRNA there is no thymine present, and in the place of thymine you have the nitrogenous base uracil. And here this is the thymine, and here you have the adenine present. So this replacement leads to the change in the amino acid that is produced. The normal hemoglobin that is produced is glutamic acid, whereas in the case of the person who is suffering from sickle cell anemia, in the place of glutamic acid, some other amino acid is produced, and that some other amino acid is valine. So this valine is responsible for the person showing the RBCs which are sickle shaped, leading to the disease sickle cell anemia. So here in the case of normal hemoglobin, the individuals will be having like this, and in the case of people suffering from sickle cell hemoglobin, the hemoglobin looks like this. So this is once again the recessive thing, homozygous recessive, and these people, they develop this disease sickle cell anemia. Carriers won't show any symptoms. Now let us see sex linked disease, that is color blindness. We have seen about color blindness in our earlier module also, but let me just quickly recapitulate what we have seen in earlier module. It is a genetic disease, sex linked disease, in which the individuals will not be able to distinguish between red color and green color. And this affects more males than in the case of females, because in the females there are 2 X chromosomes present, and in the males there is only 1 X chromosome present. So if that particular X chromosome which males are having, if they happen to inherit the gene of color blindness, then the males will show the disease of color blindness. Now here this is how the color blindness is inherited. This is a normal female and this is a colorblind male, and this is the gametes which are produced by the female and the gametes produced by the male, and this is the possibility of the F1 generation, that is normal but carrier female and the normal male. Whereas here in the second inheritance you see that you see that the carrier female, and here this particular person is the normal male. So when this happens, this X goes into the male, and then you have one male who shows the symptoms of color blindness. So this is how the color blindness is inherited. Children, these kinds of problems you will get in ISC paper also. So you may be getting various combinations, like what happens when there is a marriage between a carrier female and a normal male, or maybe a normal male and a colorblind male. What kind of inheritance is seen in F1 generation? You have to show this in pedigree chart, and you have to explain what are the various percentage of the children having this disease in the F1 generation. So let us quickly summarize what we have seen. In today's class, we have seen genetic disorders, phenylketonuria, thalassemia, color blindness, and sickle cell anemia. Thank you, children. Stay home, stay safe. God bless you all.